Good morning to all of you students of BT first sem. Today, I'm going to discuss with you about the mitochondria, which is given in your cell biology syllabus. So, let's get started now. So, we will discuss about mitochondria today. Mitochondria plays a important role in generation of metabolic energy in the eukaryotic cells. You have already learned in class 12 that, and also bit of in your biochemistry, that this mitochondria plays an important role in generation of metabolic energy in eukaryotic cells, and they are responsible for most of the energy, useful energy, which is derived from breakdown of carbohydrates and fatty acids. So the carbohydrates and the fatty acids, they are going to be breaking down within the cell via glycolysis, Krebs cycle, electron transport chain, and they will be converted into ATP by the process of oxidative phosphorylation, as we know. Now, most of the mitochondrial proteins are translated on free cytosolic. They are transported or translated on free cytosolic ribosomes, and then they are imported into organelles by specific targeting signals. So what does it mean? It means to say that most of the, again I repeat, it is saying that most of the mitochondrial proteins are translated. Translation is the process where the uh, mRNA will be used as a template for the process of protein synthesis. So here the mitochondrial proteins are first translated on free cytosolic ribosomes. That means the ribosomes which are found on the cytoplasm, and then these proteins are imported to the mitochondria by specific targeting signal molecules. So the proteins which form the mitochondria, which perform the functions of mitochondria, they are first translated by the cytosolic ribosomes, the free ribosomes, and then these proteins are allowed to import to within the mitochondria with the help of specific targeting signal molecules. In addition, mitochondria are unique among the cytoplasmic organelles in that they contain their own DNA. Now, as endosymbiotic theory, we have learned in our junior classes that mitochondria was found to be an uh, organism. Okay, it is an organism which have come into contact with our eukaryotic cells, and by the process of endocytosis they get inserted, and during the course of evolution they become a part of the cell as a cell organelle. So that's why mitochondria have their own DNA. Similarly, chloroplast is also one of the primitive organelles which have come into contact with the plant cells and become a part of the plant cells. So mitochondria are unique from other organelles because they have their own DNA, and that DNA will encode your transfer RNA, ribosomal RNA, and some mitochondrial proteins. Okay, so mitochondria have their own DNA, and that DNA will be responsible for coding the tRNA, which is the transfer RNA needed to transfer specific amino acid for the purpose of translation process, rRNA, which is called ribosomal RNA, which will involve in formation of ribosomes, and some mitochondrial proteins. Assembly of mitochondria does involve proteins encoding by their own genomes and translated within the organelle, as well as the proteins encoded by the nuclear genome and imported from the cytosol. So some proteins are produced within the mitochondria. Okay, they have their own ribosomes and they translate to form that proteins. Okay, but there are some proteins which are encoded by the nuclear genome. Some of the proteins of the mitochondria are encoded by the nuclear genome. After translation, that proteins has to be imported from the cytosol to the mitochondria via specific targeting signals. With the help of targeting signals, that proteins after translation will be imported to the mitochondria. So all the proteins are not synthesized by the mitochondria. Some of the proteins are encoded by the nuclear genomes that has to be transported into the mitochondria. Other proteins are produced within the mitochondria as they have their own DNA that will encode some of the mitochondrial proteins along with tRNA and rRNA. Okay. Now let us see about the organization and function of mitochondria. Well, mitochondria are surrounded by a double membrane system. You have seen the structure many times of mitochondria. They are surrounded by a double membrane system. They have a inner membrane and outer membrane, and between the inner membrane and outer membrane there is a intermembrane space, right? And within the inner membrane there are inner folds like structure which are known as cristae, and this cristae like structure extend into the interior or matrix of the organelle. So again I repeat, mitochondria are surrounded by a double membrane system. They have an inner membrane and outer membrane, and between the inner membrane and outer membrane there is a space called intermembrane space. Inner membrane have tiny projections, tiny fold like structure called cristae, which extend to the interior of the mitochondria. Each of these components, inner membrane, outer membrane, the cristae like structure, each of these components play a distinct functional role, with the matrix and inner membrane representing the major working compartments of the mitochondria. So the major function of the mitochondria is performed by the inner membrane, okay, and the matrix. That means the interior part of the mitochondria perform the main functions. Okay, so this is the structure of mitochondria, which I have already said. Look at the crystal-like structure which have fold inside of the inner membrane. There is a outer membrane also, as you can see, and between the inner membrane and outer membrane there is a intermembrane space. So this is your intermembrane space. As you can see, these are the crystal-like structure which are uh, within the matrix, within the internal side. Oh, okay. And most of the work occurs within the matrix. The FO F1 particles, F not F1 particles, as you know, you have already studied on class 12 also. They are found within this crystal like structure. Okay. Okay, so let's move to the next part. So it is saying here that the matrix contain the mitochondrial genetic system. Matrix means the inner part of the mitochondrial membrane. So matrix contain the major mitochondrial genetic system as well as enzymes which are responsible for the central reactions of oxidative metabolism. So the matrix, which is the inside inner part of the mitochondrial membrane, the interior of the mitochondrial membrane, it contains the mitochondrial genetic system, as well as it contains the enzymes which are responsible for the oxidative phosphorylation uh, that is done within the mitochondria. Well, oxidative phosphorylation takes place within the mitochondria. They use molecular oxygen during this reaction, and it is an exergonic reaction where the uh, metabolized products are actually gets converted into energy. This is the last step of your, what can I say, energy production from the carbohydrates that we are eating. Okay, so for the oxidative phosphorylation, many enzyme systems are required, and that is provided by the mitochondria here. Oxidative breakdown of glucose and fatty acid is the principal source of metabolic energy in animal cells. As we have learned in our classes, early classes, that oxidative breakdown of glucose and fatty acid is the main principal source of metabolic energy in animal cells. Okay, so to break down the glucose by oxidation, it is the main, and fatty acid also they get breakdown to get energy by the animal cells. Initial stages of glucose metabolism is your glycolysis. As you know, it occurs within the cytoplasm where glucose is converted into pyruvate. Then pyruvate is then transported into the mitochondria where complete oxidation to CO2 yields bulk of usable energy obtained from glucose metabolism, right? So the initial stage, as we know, that glucose is metabolized by glycolysis, the 10 step reactions which occurs within the cytosol where glucose is converted into pyruvate. This pyruvate will be transported into mitochondria where complete breakdown of oxidation of um, will occur, which will yield a bulk of usable energy obtained from the glucose metabolism. Now this involves initial oxidation of pyruvate to acetyl coenzyme A. So the pyruvate that we obtain from glycolysis, it will be converted, it will be oxidized into acetyl coenzyme A, which will be further broken down to CO2 via citric acid cycle. And with the help of this same technique, same process, oxidation of fatty acid also occur, which will yield acetyl coenzyme A, which will be similarly metabolized by the citric acid cycle within the mitochondria. So generally, as we know, that metabolism of carbohydrate takes place within the cytoplasm and then within the mitochondria, where the glucose is break down to form pyruvate by the process of glycolysis. Then this pyruvate that we get, it is oxidized to form acetyl coenzyme A, which will be further broken down to CO2 via the citric acid cycle. Similarly, fatty acids are also oxidized to yield acetyl coenzyme A, which will be similarly metabolized by the citric acid cycle within the mitochondria. So now we need the enzymes that are involved in the citric acid cycle. So the enzymes of the citric acid cycle, which is located in the matrix of mitochondria, are the central players in the oxidative breakdown of both carbohydrates and fatty acids. So this should be remembered that the enzymes of the citric acid cycle, which are located in the matrix of mitochondria, are the central players in the oxidative breakdown of both carbohydrates and fatty acids. Okay. So this is the structure, general structure, as we know, see that the pyruvate that we obtained from glycolysis through the process of oxidative citric acid cycle, they are first going to convert to acetyl coenzyme A and then to CO2. The fatty acid also makes the same thing to yield out, and from the products of electron transport carriers which will be involved in electron transport chain further to produce energy. Okay. And this is all function are performing within the mitochondria actually. Okay. Now more thing has been given that after the formation of acetyl coenzyme A, which is metabolized by the citric acid cycle, okay, which will be further broken down to form CO2, right? Then during the formation of CO2 from acetyl coenzyme A, it is coupled with the reduction of NAD plus and FAD plus to NADH and FADH2. So when oxidation of acetyl coenzyme A occurs to form CO2, at the same time it also reduces the NAD plus to NADH. It also reduces the FAD to form FADH2, right? And most of the energy derived from oxidative metabolism is then produced by the process of oxidative phosphorylation, which takes place in the inner mitochondrial membrane. Okay, it takes place within the inner mitochondrial membrane. So during the process of conversion of acetyl coenzyme A to CO2, NAD plus and FAD get reduced to form NADH and FADH2. Now this NADH and FADH2 are actually they transfer the electrons, okay, through the process of electron transport chain. The high energy electrons from NADH and FADH2 are now transferred through a series of carrier molecules in the membrane to the molecular oxygen. So the high energy electrons from NADH and FADH2, they are transferred through a series of carriers in the membrane to molecular oxygen. Now the energy derived from these electron transfer reactions is converted to potential energy stored within in a proton gradient across the membrane, which is then used to drive the ATP synthesis. So in a repeat, the NADH and FADH2 that we get from this NADH and FADH2, high energy electrons that is present within the NADH and FADH2 will be transferred through a series of carriers, right? Because reduction means, removal of oxygen, addition of hydrogen, or addition of electron. So the high energy electrons from NADH and FADH2 will be transferred through a series of carrier molecules, through the series of carrier complexes as synthesized by the mitochondria within the membrane to molecular oxygen. Now the energy derived from these electron transfer reactions, which is together called as electron transport chain, this energy that is derived from these electron transfer reactions will be converted into potential energy. That energy will be stored in a proton gradient across the mitochondrial membrane, which is then used to drive the ATP synthesis. So the inner mitochondrial membrane thus represents the principal site of ATP generation, and this critical role is reflected in its structure. So in this way, the inner mitochondrial membrane plays an important role, which is the important site for ATP generation. As I have said that how ATP is produced, okay, by breakdown of the carbohydrate and fatty acids. Okay, now it is saying that first about the inner mitochondrial membrane. Its surface area is substantially increased by folding into cristae. So if you look at the structure of the mitochondrial inner membrane, its surface the surface area of the inner mitochondrial membrane has been increased because of that tiny projections folding like structure called cristae. In addition, the inner mitochondrial membrane contains unusually high percentage of proteins which are involved in the oxidative phosphorylation as well as in the transport of metabolites between the cytosol and mitochondria. So it is saying that why inner mitochondrial membrane are the site of ATP generation? It's because first its surface area is quite large because of that crystal like structures. Secondly, most of the proteins, about more than 70% of the proteins which are involved in the oxidative phosphorylation as well as transport of metabolites, that is the pyruvate and fatty acid from cytosol to mitochondria, are done by the inner mitochondrial membrane proteins, right? Otherwise, the inner membrane is impermeable to most ions and small molecules, a property critical to maintain the proton gradient that drives oxidative phosphorylation, right? So that's why the inner mitochondrial membrane is quite important for ATP generation. The whole process, if we summarize, it's first, as we know, that first the glucose, if we go for carbohydrate metabolism, the glucose is first metabolized to form pyruvate, which is then further oxidized, to give acetyl coenzyme A, which will be broken down to CO2 via citric acid cycle. And then during the oxidation of acetyl coenzyme to CO2, there will be reduction of NAD plus and FAD plus to NADH and FADH2. Electrons are carried by the NADH and FADH2, right? And they are transferred through a series of carrier molecules, through a series of carrier complex within the membrane of mitochondria to molecular oxygen. Now the energy derived from this electron transfer reactions will be converted into potential energy, which will be stored in a proton gradient across the membrane, which will be then used to drive the ATP synthesis. So that's why the inner mitochondrial membrane is the main principal site of ATP generation, and its structure represents that role actually. As we have seen that the inner mitochondrial membrane have a large surface area because of that folding like projections, crystal like structure. In addition, most of the proteins which are involved for oxidative phosphorylation, which are involved for transport of metabolites, are found within the inner mitochondrial membrane. Otherwise, the inner mitochondrial membrane is quite impermeable to most ions and small molecules. Okay. Now let us see about the uh, here the overview complex of that proteins has been shown in the inner mitochondrial membrane. The complex 1, complex 2, complex 3, complex 4, complex 5. So which are important for oxidative phosphorylation, which are important for the electron transport chain. This is the complex 2, which are involved in the electron transport chain. Many proteins are involved here, and they are all found within the inner mitochondrial membrane, which are performing this function, okay, to generate ATP. Now, in contrast with the inner mitochondrial membrane, the outer mitochondrial membrane is freely permeable to small molecules. That means within the outer mitochondrial membrane, we can easily transport the small molecules because it is permeable. This is because it contains small proteins, carrier proteins called porins, which forms channels that allows free diffusion of molecules smaller than 6,000 daltons. So the outer mitochondrial membrane is quite permeable to most of the proteins, because most of the molecules, because the outer mitochondrial membrane have channel proteins called porins, which allows uh, smaller than 6,000 dalton molecules to easily transfer. into the mitochondrial membrane. The composition of inner intermembrane space is therefore similar to cytosol with respect to ions and small molecules. The intermembrane space, which is found between the inner mitochondrial membrane and outer mitochondrial membrane, is quite similar to the cytosol, which has ions and small molecules. Consequently, the inner mitochondrial membrane is the functional barrier to the passage of small molecules between the cytosol and matrix and maintains the proton gradient that drives the oxidative phosphorylation. Because that inner mitochondrial membrane is quite selectively permeable, it is acting as a wall for the passage of small molecules between the cytosol and the matrix. That's why it maintains that proton gradient. It maintains that proton gradient that drives the oxidative phosphorylation process. Okay. So inner mitochondrial membrane is the main functional barrier. Outer membrane is allowing the molecules because it has channel proteins called protins, which allows less than 6,000 Dalton proteins molecules to transport. And the inner mitochondrial membrane is quite acting as a barrier for the passage of small molecules between the cytosol and the matrix is the inner part of the mitochondria, which maintains that proton gradient that drives the oxidative phosphorylation. Okay, next we will study about the genetic system of mitochondria. Now, about the genetic system of mitochondria, what it is saying here, let us see. It is saying that mitochondria contain their own genome, which is separate and distinct from the nuclear genome of the cell, as I have said. And I have also said that mitochondria are thought to have evolved from a bacteria that develop a symbiotic relationship in which they live with larger cells, that is endosymbiosis. So according to the endosymbiosis theory, mitochondria was thought to be a bacteria. Okay, and it has been observed that the genome of the mitochondria is quite similar with the bacterium called Rickettsia prowazekii. Okay, Rickettsia prowazekii. So it has been a little bit confirmation has been done that mitochondria may have evolved from the bacteria Rickettsia prowazekii, which have entered to the cell, eukaryotic cell, and perform an symbiotic relationship with that larger cell. These hypotheses have recently been substantiated by the result of DNA sequence analysis, which revealed the striking similarities between the genomes of mitochondria and of the bacterium Rickettsia prowazekii. That means DNA analysis has been done between mitochondria and between the bacterium Rickettsia prowazekii, and strikingly, scientists have discovered that the genome of mitochondria is quite similar with the bacterium of Rickettsia prowazekii. From this, we can conclude that mitochondria have evolved from Rickettsia prowazekii, which is a bacterium. Rickettsia are intracellular parasites which is like mitochondria and are only able to reproduce within the eukaryotic cell. According to the properties of the Rickettsia, it has been confirmed or more confirmation has been done that these Rickettsia are intracellular parasites and they can only able to perform their reproduction when they get entered to the eukaryotic cells. Consistent with their similar symbiotic lifestyles, the genomic DNA sequence of Rickettsia and mitochondria suggests that they share a common ancestor from which the genetic system of present day mitochondria evolved. Now, by observing all these functions and DNA analysis of Rickettsia and mitochondria, finally, scientists have concluded that the Rickettsia and mitochondria both have evolved from a common ancestor, from a common bacterium, from which the genetic system of present day mitochondria have evolved. Now, the mitochondrial genome are usually circular DNA molecules. So I will give you in the notes. You will also see here also that that mitochondrial genomes are usually circular, like those of bacteria, which are present in multiple copies per organelle. Like in bacteria, we see a circular genome. Okay, um, here also mitochondrial genome are also circular, and the genome vary considerably in size between different species. So depending on species, it varies. The genome size varies. The genome of human and most other animal mitochondria are only about 16 kb in size. Only 16 kilobases in size is observed on the human mitochondrial genome. But substantially large mitochondrial genomes are found in yeast, approximately 80 kb, and in plants about more than 200 kb. Okay, but it does not means that more genome means more proteins they will encode. Okay, it has been observed that large mitochondrial genomes, most of them have non-coding sequence which will not code any proteins. So however, large mitochondrial genomes are composed predominantly of non-coding sequence which do not appear to contain significantly more genetic information. So the genome size of mitochondria varies between different organisms. Like in humans it contains only of 16 kb, whereas in plants you can see more than 200 kb of mitochondrial genome are found. In yeast about 80 kb of mitochondrial genome is found. But most of this case, what happened, most of the mitochondrial genome are non-coding sequence, they will not code for any proteins. Okay, now let us see about the human mitochondrial genome. The human mitochondrial genome encodes 13 proteins which are involved in electron transport chain and in oxidative phosphorylation. Okay, so the human mitochondrial genome encodes 13 proteins, 13 proteins, which are involved in electron transport chain and in oxidative phosphorylation. In addition, human mitochondrial DNA encodes 16S and 12S ribosomal RNA, which is also called as rRNA, and 22 types of transfer RNA, which is called tRNA, which are required for translation of proteins encoded by that organelle genome. So the human mitochondrial genome have 13, it consists of genome which will coding for 13 proteins which are involved in electron transport chain, which are involved in oxidative phosphorylation. In addition, mitochondrial DNA of human will also encode for the ribosomal RNA for the synthesis of ribosomal proteins like 16S rRNA, 12S rRNA, and also it will synthesize the transfer RNA which are needed for transport of specific amino acid within the site of ribosome by matching with the codon sequence of the mRNA and anticodon sequence of the tRNA by the process of translation. So transport RNA is also needed, transfer RNA is also needed, and which is also synthesized by the human mitochondrial genome. The two rRNA that we have seen here just now, the 16S and the 12S, are the only RNA components of animal and yeast mitochondrial ribosomes. So these two rRNA, which is 16S and 12S rRNA, are the only RNA components of animal and yeast mitochondrial ribosomes. In contrast to the three rRNAs of bacterial ribosomes, 23S, 16S, and 5S. That means you can compare this 16S and 12S rRNA with the three rRNA which are found in the bacterial ribosomes, right? That is 23S, 16S, and 5S. Recall your concept in your class 12 that 16S rRNA will actually helps to complementary bind with the mRNA, which we called as ribosome binding site or Shine-Dalgarno sequence. The 23 rRNA, 23S rRNA is involved in peptidyl transfer reaction, where the main a peptide bond formation occurs between the two amino acids carried by the tRNA in the A site and in the P site. Okay, so these all are RNAs that is found within the bacterial ribosomes. They are quite comparable with this 16S and 12S rRNA found within the mitochondrial genome. That means they will perform the same function as these are doing. Okay. Plant mitochondrial DNA, however, also encode a third rRNA of 5S. Now, within the plant mitochondria, they are other than 16S and 12S, they also encode 5S rRNA. Mitochondria of plants and protozoans also differ in importing and utilizing the tRNA encoded by the nuclear as well as the mitochondrial genome. So the mitochondria of plants, the mitochondria of protozoa uh, also differ in their function according to their importing of tRNA and utilization of tRNA, right? So this is the main genome of the this is the main mitochondrial genome. I am providing you a color picture now. This is a black and white. I'm Wait for a minute. I will provide you the genome of the mitochondria in color picture. Okay, wait. So this is the color picture of your mitochondrial genome. So where you can see different regions. We can see dark green, you can see gray regions, pink regions, and something written also here. What these represent, we will see here at the bottom. Well, this is the structure of mitochondrial DNA taken from Google source. Uh, well, what it is written here: mammalian mitochondrial DNA, also called as mtDNA. It is a double-stranded circular DNA molecule, as I have mentioned with you before, with approximately 16,000 base pair. Total 16 kb kilo bases. Mitochondrial DNA is formed by a light strand and a heavy strand. So the human mitochondrial DNA, if you see, it has two strands. One is called light strand, and another is called heavy strand. The all the pink color that you are seeing here, it denotes the tRNA coding sequence. There are about 22 mitochondrial tRNA. Each are labeled with respective amino acid code. So here you can see all the pink regions, all the pink regions represents the region, all the pink regions that you can see here, these are the DNA sequences which will code for tRNA, which will code for tRNA. Now this one letter code represents that what amino acid that particular tRNA carry. Okay, I'm giving you that. I will be giving you in the notes in the one letter amino acid code that what type of amino acid is coded by that one letter. Okay. Otherwise, we use three-letter code of amino acids. So all the pink regions represents the tRNA that will carry the particular amino acids. Then it is saying that protein coding segments on mitochondrial DNA do not have introns and are transcribed by a single polycistronic mRNA from each strand. So there is no such intron sequence here. Just like bacteria, they they are continuous. They have one open reading frame, then next open reading frame, then next open reading frame, which will code for particular proteins. Similarly, here in mitochondrial DNA, they do not have any interrupting sequence called introns, and they are transcribed by polycistronic mRNA. That means they will have multiple cistrons, multiple coding sequence will be there, multiple open reading frame will be there, which will code for a particular protein. Okay. All protein coding sequence are marked in green color with respective gene name abbreviations. So here we have seen some abbreviations here which shows that color. Like for example, CYTB. CYTB represents that particular site gene of mitochondria will encode cytochrome B. As you can see, CYTB, it represents that here the gene that is found within the mitochondria will encode cytochrome B synthesis. Then it is saying ND. ND means NADH dehydrogenase, which is required for the electron transport chain. Um, as we can see, now there are many things. ND has been written ND6, ND5. So these are all NADH dehydrogenase coding genes that is coding by the mitochondria here. Here also you can see ND4, ND4L, ND3. Okay. Then comes CO. CO represents cytochrome C oxidase. That means um, this part where CO is written, that means that particular site of the mitochondrial DNA it will code for the cytochrome C oxidase enzyme. Okay, you can see here CO3, CO2, CO1. This represents cytochrome oxidase, right? It will code for cytochrome C oxidase actually. And there's also ATPase also, gene which will encode ATPase, okay, which is ATP synthase. Okay, so in this way the whole mitochondrial DNA is marked in this way. If I small it out, it will look like this. Okay, you can take a screenshot for writing the notes. Otherwise I will also send you the picture here also in your WhatsApp group. Okay, so this was all about the mitochondria genome, its functions, its structure. Just go through this chapter. Okay, and if you have any doubts regarding this part, you can ask me in the WhatsApp group. Okay.